So sometimes with final exam, it can be a little bit tricky to figure out what you're supposed to know, the level at which you're supposed to review things. So we're going to take a few minutes here to review some old cumulative content at the level appropriate to a final exam. One of the things we talked about in the beginning of the semester was the different organ systems. We just highlighted them. So even now, for each organ system we're about to name, you should be able to name a couple of organs and then just a couple of overarching functions. So this is a representation of the integumentary system, which is the skin and its appendages. And the skin is things like ultraviolet light protection, protection from UV light so you're less likely to sunburn and develop cancer. Protection from the environment. Okay? It's the boundary between the in and the out. Vitamin D production, temperature regulation, excretion. So we'll review a little bit more about skin later. We also looked at the nervous system in a lot of detail, the central nervous system and the peripheral nervous system. At the beginning of the semester, we said all those nerves and that brain and the spinal cord are for homeostasis. Very fast detection, fast response, homeostasis. And we just spent a lot of time talking about the muscular system. The muscular system is for movement. Muscle cells contract. But because the three tissue types are a little different, when they contract, you get a different effect. We briefly mentioned cardiovascular system 4 months ago. That's the heart, the arteries, the veins, and the capillaries. It is a transport system for moving nutrients and waste. Lymphatic and immune systems are tied to each other. The lymphatic system captures fluid and returns it and filters it along the way. The immune system is there for surveillance, to keep the bad things out. Respiratory system, that's the lungs and the trachea and the bronchi. The respiratory system is a transport system but for gases, so it's gas exchange, and we can use that gas exchange to help regulate blood pH. Digestive system, that's the intake, breakdown, absorption, and elimination of food. Everywhere along the alimentary canal, so esophagus, stomach, small intestine, large intestine, colon, pancreas, liver, salivary glands. Urinary system, kidney, ureters, urinary bladder, urethra. They filter blood and eliminate waste. And reproductive system is to make more humans, so testes, ovaries, vagina, penis, all structures of reproductive system. After we had that introduction, we started to look at the hierarchy of life. We defined life, and we talked about the characteristics of life, like movement, homeostasis, responsiveness. And we said that living things are organized from simple to most complex in this manner. We are all atoms. If we get different atoms together performing common function, those become molecules. Get different molecules together performing common function, we have organelles. Different organelles together performing common function, we now have cells. Different cells performing common function, we have tissues. There's a theme here. Different tissues together performing common function, organs. Different organs together performing common function, organ system. And if you get all those organ systems together performing common function, that's an organism. So this hierarchy is one of the themes that runs under the class all semester long. Everywhere along the way, on every exam, you've been given a set of terms or a set of things, and you had to correctly group them and organize them based on this hierarchy. So expect to do that again on the final exam. We'll go through an example in a second. The other reason why we'll test you on your understanding of this hierarchy is to give you one item and have you tell me where on this continuum it actually falls. So example, if I say liver, organ. If I say cardiovascular, organ system. If I say neuroglia, that's a cell. If I say perineurium, that's a tissue. If I said terminal cisternae, that's an organelle. So let's work an example together. On the right-hand side of the screen, you can, in black, you can see the things I've listed here that I want you to organize based on that hierarchy. So when you're given a list like this, the first thing to do is work your way down the hierarchy. We know that the first simplest level is the atom. So on this list, can you find an atom? An atom can also be written as the element. There is an atom written here, an element written here, and that is calcium. So let's go one higher up. Let's try to identify molecules. Well, if you go through the list, we find two molecules, actin and thin filament. So the question in this example becomes, which one is the smaller of the two, or the more simple of the two? That would be actin. So actin comes next, and actin is part of the thin filament. So that's the next level up. Well, now we've checked the molecules off our list. Now we need to find the organelle. So go through this list and see if you can spot an organelle. Remember, that's something inside a cell. The organelle here is the myofibril. So check that one off your list. Now we need to find the cell. Can you spot the name of a cell in here? That would be the muscle fiber. Remember, muscle fiber, muscle cells are so long they're referred to as fibers. Now we need a tissue, a collection of cells. You know, the other way to think about a tissue is if you see something that's connective tissue, nervous tissue, epithelial or muscle tissue. So the only thing on this list that fits the category of being a tissue is something that's made of connective tissue. And that would be the epimysium. And lastly, the last thing we're left with is the organ. And the organ is the biceps brachii. So I encourage you to go into your old exams, remember I've posted them as practice tests, and pull some of those example questions. Also make up your own. Go to each organ system, go through your notes, write down a molecule, a cell, a tissue, an organ, and throw them in different orders and see if you can piece it out. Once we moved past hierarchy, we went into the subject of homeostasis. Remember, the ability to maintain a relatively constant internal environment with respect to changes in the external environment. Or, as Ms. Klein says, the ability to keep the inside about the same even though the outside is changing. Now, all homeostatic mechanisms have the same components, and we'll look at those in a second. But these components have been showing up all semester long. They should sound very familiar. And we can use these components in one of two ways to maintain that internal environment. Most of the time, we are going to maintain our internal environment with negative feedback, meaning the response turns the system off. Every now and then, we got to get drastic, and we'll use the opposite, positive feedback. With positive feedback, you get more and more and more of a response. So this diagram here are the components of I just called it a reflex art, but guess what it is? So this is the layout of the nervous system. This is the layout of the endocrine system. This is the layout of a reflex. These are the components of homeostasis. A specialized receptor detects a stimulus or a change in a variable. That information is sent along an afferent pathway to a control center for integration and problem solving. The control center determines what has to happen, sends that information out along an efferent pathway to an effector for a response. And remember, if we have a situation where our response stimulates more signals, so we get more and more and more response, that's positive feedback. I gave a few examples, like estrogen in the beginning of the female's ovarian cycle, oxytocin during labor, the vascular spasm, part of the clotting mechanism in humans, But because positive feedback can get carried away, we don't have that many examples of it. Generally, to maintain homeostasis, we actually don't want more and more and more. We want more, and then we want to stop. Or we want less, and then we want to stop. So the vast majority of the mechanisms are indeed negative feedback. The response would turn this off. So your blood glucose is a little low. The components fire. You make a little blood glucose. The moment your blood glucose levels get back to normal, you turn the system off until you need it again. So not only should you know the components and be able to differentiate positive and negative feedback, if given an example you've never seen before, you should be able to figure out, is this negative feedback? Is this positive feedback? Or is it not homeostasis at all? After hanging out with homeostasis for a while, we started talking about cells, and we started with the cell theory. And I remind you that since the 2nd grade, you've been learning that the cell is the unit of life. And that hasn't changed. But we did talk a little bit about why they're small. And basically they're small because their size is limited. The bigger a cell gets, the more nutrient and waste it needs and produces, and the more cell membrane it needs to move the nutrients and waste. Well, when a cell gets really big, guess what? It doesn't have enough cell membrane on it anymore to move its waste and nutrients. I call that surface to volume ratio. So cells can never get really big because their surface to volume ratio limits their size. They have to stay small. We talked a little bit about the process of specialization. You know, we all started out as an egg and sperm that made friends, a fertilized egg that divided into a ball of cells. Those were stem cells, undifferentiated cells. And to this day, throughout the semester, we've talked about stem cells, some still being in the brain, some being in the umbilical cord, some potentially and possibly still being in ovaries. The deal is that those cells, those undifferentiated cells, there's even one in the bone marrow we talked about. Anyway, those undifferentiated cells go through a process where they become a specialized cell. Once they do that, there's no turning back, and they grow up and they know what they need to do. So for example, there's a stem cell in the bone marrow, and that stem cell can become a red blood cell or any one of 5 plus white blood cells, or even a platelet. When it's the stem, it's called a hemocytoblast. Don't write that down. But a hemocytoblast is an undifferentiated cell. It just knows it has to become some kind of blood cell. Depending on the hormone or the trigger that you put on it, it will specialize to a specific population. So once the cells become very, very specialized, their job is defined. They're not generic anymore. They grow up, they know what to do. Part of specializing means your structure changes so that your function can change. And with the cell theory, we also started talking about the fluid mosaic model. That's a phospholipid bilayer with proteins and cholesterol. And what you should understand at this stage in the game is that the structure of the cell's membrane helps determine how it transports molecules. So how do cells transport molecules? Let's go to the one that says passive first. Passive transport is what we get when a cell moves a molecule with a gradient, and that molecule is pretty easy to move. It's not that big, and it's soluble across the membrane. So passive includes things like, not that one, save that for later, diffusion, facilitated diffusion, filtration, and osmosis. Well, sometimes a cell has to move something really, really big, or it has to move it against a concentration gradient. If that's the case, we need to invest some energy, and we're going to have to use active transport. So that sodium-potassium pump that we first met with action potentials, that is an active transporter. That pump has to use ATP to force those ions to move. We also talked at length about osmosis. We talked about it in lecture. You've been tested on it. I said it never goes away. Even played with it in the lab. So osmosis, remember, is the diffusion of water across the cell membrane. And osmosis occurs because a cell cannot stop water from moving across its membrane. Water is one of those things that's permeable. So, if a cell is sitting in an environment that has the same amount of solute, or the same amount of stuff, we would say the cell is in an isotonic environment, and for every one water molecule that goes in the cell, exactly one water molecule leaves. So it's balanced. If a cell is sitting in a hypotonic environment, that's a cell sitting in an environment with less stuff. So there's more stuff inside the cell. That water is going to go from the environment into the cell, and the cell might actually explode or lyse. It'll definitely swell up. If you put a cell in a hypertonic environment, the opposite is true. This environment has more stuff than the cell does, in which case water leaves the cell and goes toward that stuff in the environment. And this cell would crenate, or lose water, over time. You will be asked about osmosis on the final exam. So you want to look at this, you want to look at the old integrity reviews, you want to look at the PowerPoints, you want to get the hang of osmosis. So no matter what situation I give you, you can work the problem through and see what's happening. Then the day came, and it was the day that I put that tissue sign up on the board, and then our lives changed forever. We said that in the world of the human body, there are 4 categories of tissues: epithelial, connective, nervous, muscle. So let's highlight these first 2 categories. Remember, the purpose of the final exam is to see what kind of big picture stuff do you still know, or to illustrate what's important. So as you're going through, imagine if you were the teacher, what would you want your students to remember? All right, so epithelial tissue had apical and basal surfaces and a basement membrane. And we pointed out basement membranes all semester long. Remember me saying, oh, if the cells are close together, they stay dark, and they're in contact with a space, that's epithelial tissue. So epithelial tissue is very regenerative in that it divides easily. It is avascular. The blood supply is not in it, it is under it. And function-wise, this is protection, if it's stratified, filtration for simple squamous, gas exchange for simple squamous, absorption in the gut, simple columnar, absorption in the kidney, simple cuboidal. Connective tissue, completely different category. So remember, this is cells in a non-living extracellular matrix. In class I said, this is cells in some stuff that they made. That stuff is background material called ground substance and fibers, and the fibers were proteins, including collagen. There are categories of connective tissue. And so the big ones were either loose or dense, but connective tissue proper. In that category was adipose and areolar, and ligaments and tendons, dense, regular, dense, irregular. We also had cartilage. Remember, we looked at hyaline cartilage and fibrocartilage a lot. Now, as far as function is concerned with connective, this is a huge category. This is everything from blood to bone. So, protection, insulation, nutrient storage, transport, shape, things like that fall under the function of connective. Now, lecture exam, you will not be given a slide, but if you were given a very basic descriptive of one of these tissues, could you figure out if it's epithelial or connective based on what you see in front of you? If I described something that had a lot of cells that was regenerative, could you pick out that that was epithelia? If I talked about digestion in the, or if I said gas exchange at the lung, would you know that that's simple squamous? Also for connective tissue, if I said, you're looking at a microscope and you're looking at, certain fibers, certain structures, certain ground substance, could you tell if that were blood or bone or adipose or areolar. That kind of stuff. The first organ system, we learned the fact that there are two layers, epidermis and dermis, and that each had sub-layers. We learned that there's different cell types, and those different cell types have different function. And we learned that the integumentary system in and of itself has appendages like hair follicles and sweat glands, and that each of those has a different function. In fact, the integumentary system physical protection by being a So here are some. Looked at the epidermis, we would see keratinocytes, there were a couple of others we mentioned, but focus on these guys. So, effect that would have. Tissue. Dermis as a layer is connective tissue. Then we also looked at these sub-layers. Now, I'm not gonna test you heavily on the sub-layers. You should be able to put them in the correct order, however. You should also know stratum basal is where all the division takes place. Stratum corneum is sitting on top. And in the skeleton, all the macro stuff in the line. Remember, the skeletal system had three populations of cells in there. There were osteoblasts, and I said in class, osteoblasts build bone. We had osteocytes. They are mature osteoblasts. Their job is to maintain the matrix. And then we had osteoclasts. They dissolve matrix. Osteoblasts and osteoclasts have to work together to keep bones a certain shape. So you definitely know those three cell types and what they do. As far as structures are concerned in the skeletal system, you should know these terms. Perichondrium is the tissue on the outside when it was cartilage. Once that became a bone, we called it that same tissue periosteum. When periosteum goes inside a bone, we called it endosteum. Lamella are layers of matrix. Lacuna are cavities that the cells sit in. And collagen is the fiber. Hydroxyapatite is the calcium. And that's it. Don't go into more detail than that. Hormones, very important. Calcitonin and parathyroid hormone are both used to regulate blood calcium. If blood calcium levels are high, calcium is so important in the body that we want to store it for another day if we have extra. So when calcium levels are high, we use calcitonin to stimulate osteoblasts and to put that calcium into bone matrix and keep it. If calcium levels are low, we use parathyroid hormone to stimulate osteoclasts. They pull calcium out of the matrix. we talked about growth hormone and the fact that growth hormone stimulates growth, right? And how growth occurs at an epiphyseal plate. So we talked many times, I've asked you many times, epiphyseal plate, that's cartilage, that bone can grow. Epiphyseal line, that is now bone, that bone will not grow. We talked a little bit about the sex hormones and just that they change the shapes. Left about nerve. If I gave you the stages of an action potential, could you put them in correct order? Do you understand depolarization and hyperpolarization and repolarization? Do you understand what would happen if sodium entered a cell or if potassium left a cell? Definitely understand neurotransmitters in a synapse. If I gave so again, so for spinal cord, you should be able to differentiate gray matter and white matter and their functions. For the parts of the brain, the 4 parts of the brain and their function. So again, can you identify the 4 parts of the brain? Do you know what the general functions of those 4 parts are? Can you differentiate between white matter and gray matter? Can you describe the function of the spinal cord of the dorsal versus the ventral parts? Remember, dorsal, sensory. Ventral, motor. In the world of the peripheral nervous system, we talked about cranial nerves, spinal nerves, and the structures of a nerve. So can you describe the structure of a nerve using the terms epineurium, endoneurium, perineurium, and fascicle? Do you understand that the fascicle is a bundle of cells, and these 3 things listed here are tissues? Do you know the regions of the spinal nerves? Guess what? It's the same as the vertebral column. Cervical, thoracic, lumbar, sacral, coccygeal. We talked a lot about the cranial nerves, but there's one cranial nerve that's famous. It carries parasympathetic fibers. It is sensitive. In the world of the autonomic nervous system, we compared and contrasted the anatomy and the function of sympathetic and parasympathetic. Sympathetic, fight or flight. Parasympathetic, housekeeping. So sympathetic is thoracolumbar, flight or fright. Short preganglionic but long postganglionic. Splanchnic and collateral were words that went with this nervous system. The neurotransmitters are norepinephrine and acetylcholine. Parasympathetic, completely different set of words. It is craniosacral. It is for housekeeping. Long preganglionic, short postganglionic. Its neurotransmitters acetylcholine. But most of the organs in the body have dual innervation, which means they have sympathetic and parasympathetic fibers to them. And everyone agreed with me that that makes sense. If you speed the heart rate up, you'd like to be able to slow it back down. So again, dual innervation means neurons of both to that organ. Now, that's the highlights of the old stuff. Of this old stuff and these subjects I've mentioned here, you should have a good sound understanding of them. If you're not sure while you're studying if a level of detail is appropriate or not, just send me an email and say, Miss Klein, do I need to reread or relearn X? And I'll tell you. I'll guide you. It's hard enough. You don't have to be on this final exam review on your own here. I can tell you with the muscular system, pretend like you're having an exam on the muscular system. Use your course objectives for the muscular system. Write out everything you know. Feel free to email that to me so I can make sure you have all the correct content. So study muscular system using your course objectives like you're having an exam on it. Then use this PowerPoint and this review as a guide for the level of the old stuff. Go grab your old exams as the practice exams and play with them multiple times. You won't necessarily see the same questions. However, the more practice you get with the contests, the better you'll become with it.